Hello and welcome back to Microbial Concepts. So in this video we are going to discuss about genetic code and its 9 characteristics, of which its Wobble hypothesis is also there. So this video is going to be a bit lengthy, but I'm trying to explain everything in this one video, so it will be helpful to you and you will get your concepts clear just watching by this one video. So do watch this video till the end and do subscribe to my channel and hit the bell icon so that you get notified on each new video that I upload. So let's start with the first, that is, what is genetic code? Okay, so genetic code is actually the relationship between, you can say, DNA or RNA sequence and the corresponding protein that is formed after translation. So that is the genetic code. Okay, so you know the central dogma now. You must have studied the transcription and then translation, right? So this is how the information is encoded within DNA, and RNA is then translated to the sequence of amino acid, that is the protein that is synthesized. So there are a set of rules by which this particular information is first transcribed to mRNA from DNA, and then the mRNA is read in translation to uh, encode particular protein, right? Specific amino acids. So chain of specific amino acids gives us a particular protein, right? So that is translation. So you must have studied now transcription and translation in detail, but here we are going to focus on the genetic code which is involved in the translation. Okay, so I'm not going to uh, refer here or revise here to the transcription because I hope you know that very well. We are just going to focus here about translation. Okay, so this genetic code which we use on, which is there on the mRNA to read and then code for proteins, it is actually of, it is a sequence of nucleotides. Okay, so 3 nucleotides in that sequence, we term them as a codon. Okay, so a single codon has 3 nucleotides. Okay, so I will teach you how to read a particular sequence. Now each codon which is present on the mRNA, or each code, it corresponds to a specific amino acid, or it can serve as start signal, that means methionine, that is AUG, it is a start signal in the translation, right? Or it can be a stop signal, that is termination of protein synthesis or translation. So it depends on what particular code it is. So you can see here, this is the central dogma. So a gene from DNA which is transcribed, we get mRNA, and on translation of mRNA we get a particular protein. Okay, so this is the sequence of a specific chain of amino acid which gives us a specific protein. Okay, now you can see here, after transcription we get a gene sequence. Now how to read this gene sequence? So always there will be a start signal for translation, that is AUG. It will initiate the translation. Okay, so after translation, this is the triplet codes. Okay, we have learned that a codon has 3 nucleotides. Okay, so once we have read this particular code, we will read the next code as GUG. We will not read GGU. Okay, there are no overlappings, right? So after AUG, once it is used, forget about this code or this codon. You will go towards the next code, that is GGU. Okay, then UUG, then AGC, and so on. So this is how we read the sequence of mRNA sequence for translation, and it always starts from AUG. Okay, so this particular gene, okay, the gene which is, or the sequence which is after AUG, that will only code for the protein, right? Now here you can see, this is the mRNA transcribed code. So this is the code, that is AUG. It will code for methionine. Then GUG will code for valine, and then GGA will code for glycine. Okay, so the tRNA will come, it will have the anticodons. Those anticodons will read the code on the mRNA, and if it matches, then it will code for the particular specific amino acid. Then after that, the second tRNA comes. Now it is carrying another amino acid. If it matches to the codon mRNA, then this particular amino acid binds to the methionine and the tRNA, empty tRNA lives. That is how transcription takes place. Translation takes place. And after completing the translation, what we get a sequence of amino acids. That is nothing but a particular protein. Now about the characteristics of genetic code. So there are around 9 characteristics. So we will study them one by one. So first is the codon, or the code is triplet. Okay, that is what we term them as codon. So code is the triplet. This is the first characteristic. So each mRNA codes that mRNA codon that specifies an amino acid in the polypeptide chain. It consists of 3 nucleotides. So it is termed as triplet code. Now second is the code is almost universal. That means if you take a code or codon, that is UUA, so it is coding for phenylalanine, and then it is same in the prokaryotes and eukaryotes. Okay, so there are few exceptions. So it is said almost. So almost all organisms they share same genetic language. So in case of eukaryote, UUU will code for phenylalanine, and also in prokaryotes it will code for phenylalanine. Okay, so that is about the code is universal. That means in case of prokaryotes and eukaryotes, almost all organisms they share same genetic language. There are few exceptions, so the word we use here is almost. Then the third is the code is non-overlapping. Okay, that means you will read it as UAUG and then CAU. You will not read as UGC, or you will not read as GCA. Okay, codes are not overlapping. So non-overlapping code means that the same base in the mRNA is not used for different codons. Okay, same base is not used for different codons. Each nitrogenous base is present only in one codon. Okay, so this A is present, or it will be used in this particular codon only. You cannot use it, or by overlapping you cannot generate another codon. So 6 bases codes for not more than 2 amino acids. Okay, so here are 6 bases. You can count 1, 2, 3, 4, 5 and 6. So we will get 2 codons here, and we will get 2 amino acids. Okay, the mRNA is thus read in the successive group of nucleotides. So how we read mRNA? We read in a successive group of nucleotides. That means 3 nucleotides, which gives us a 1 codon. Okay, so if the sequence is AUG CAU, then it represents 2 codons, that is AUG and CAU. So these are the first 3 characteristics. Now next is fourth. The codon is comma-less. That means there are no commas in between any nucleotides. So there are no punctuation between any 2 codons. So after one amino acid is coded, the next amino acid is coded automatically by the 3 next nucleotides. The mRNA is read continuously considering 3 nucleotides at a time. Okay, so that means you can see this is AUG is the start codon. So this start codon position ensures that this is the frame for the translation that is chosen. Okay, so you will read AUG and then AUC, but you can't read GAU. Okay, so the tRNA or the ribosomes, when the translation is going on, it will not read it as GAU. This is an overlapping codon, and this is the wrong way. Okay, so we always read mRNA in a continuous manner, considering the 3 nucleotides at a time. So first 3 nucleotides code for methionine, so we will forget that. We will move towards the next 3 nucleotides. Okay, we will not think about AUG again, because it will cause overlapping, and that is not how the translation goes. So the mRNA is read continuously considering 3 nucleotides at a time without skipping any nucleotide till the stop codon is reached. Okay, so you can see here in second image, this is the start codon, So during translation, it has coded for methionine and terminus. Then this coded for another amino acid, this coded for another amino acid, and this is the stop codon UAA. Okay, so this will tell the uh, translation system to stop the translation now. Okay, and this is the polypeptide that is now obtained, which will give us a particular specific protein, right? So this is how the translation goes on. So there is a very important critical role of start codon and stop codon, because that is the gene sequence that has to be transcribed. Okay, so fifth is the code is non-ambiguous. So it means that non-ambiguity about a particular codon. It means that there is no non-ambiguity about a particular codon. So a particular codon will always code for a same amino acid, and it will never code for two different amino acids. Okay, so this code will always code for a particular amino acid, and it will never code for two different amino acids. Okay, so there is an exception that AUG and GUG. Okay, this is just the exception that AUG and GUG, these are both the codons which code for methionine as initiation codons. So there are two initiation codons, AUG and GUG, but mostly you will find that AUG is one which we refer during our academic years. Okay, but GUG also codes for valine. Okay, so this is just the exception here. Then sixth is the code is degenerate, with few exceptions, that is AUG codes for methionine and UGG codes for tryptophan, because more than one codon codes for each amino acid. Okay, that means multiple coding is called degeneracy. Okay, so I will explain you. So there are particular patterns of degeneracy. So when the first two bases are identical, the third base is U or C, that is pyrimidine, and the code always codes for the same amino acid. For example, you can see here, first example is where we have UUU and UUC. Now this specifies phenylalanine, and CAU and CAC, this specifies histidine. So the third base here, that is U and C, these are pyrimidines, and A and C, they are identical. Okay, same in case of UUU and UUC. You can see the first two bases are identical in both of these codes, that is UU in this and UU in this. Just the third nucleotide is different, that is uracil in first and cytosine in the second. And that's the reason why they specify for phenylalanine. Same in this case, CA, that is histidine, cytosine and adenine, they are identical in both of these codes, and just the third nucleotide, third base is different, and thus it specifies for histidine. That means you get two codons which specifies for a particular amino acid. Why? Because the third nucleotide is different. Now in second case, when the first two bases they are identical, the third base is A or G. That means if it is purine, then they code for same amino acid. Okay, for example, again you can see UUA and UUG. So here first two bases are identical, that means they are uracil. The third base is here adenine, and here it is guanine, so it codes for leucine. In this case, AAA and AAG, so you can see here first two bases are identical, but the third base here is A and here it is G. That means they are purines, and thus they will code for lysine. Okay, so this is the sixth characteristic of genetic code, that is the code is degenerate. That means multiple coding is there, and we call it as degeneracy. So in few cases, when the first two bases are identical and the third base is either It can be, that means here in this case, if the first two bases are same and the third base is either A, G, U and C, then the same amino acid is specified. For example, in this case you can see here GCA, GCU, GCG and GCC. So first two bases, that is GC, here also GC, here also GC, here also GC. So first two bases are identical and the third base can be anything, A, U, G or C. Then all in these all cases it will code for alanine. Okay. So this is about the code is degenerate, that means multiple coding can happen. Okay. Then the fourth is the code has polarity. Okay, so if a gene is to specify the same protein repeatedly, then it is essential that the code must be read in a fixed direction and the code must have polarity. Okay, that means reading the gene in a proper direction is very important if that same gene has to specify same protein all the time. Okay, why? See here, the code for example is UUG. So when it is read in this direction, that is UUG, it codes for leucine. But when we read it in opposite direction, that is GUU, then it codes for valine. Okay, thus we follow the rule that the mRNA is always read in direction 5 prime to 3 prime. So we read codes always in this direction. Okay, but in exam you may get a tricky question where the mRNA is in the diagram, it is placed opposite, that means it is from 3 prime to 5 prime direction. So you always have to see the direction of the mRNA and then start reading it. Okay, so remember that the code they has polarity. Okay, if the code is read in opposite direction, it would specify two different proteins since the codon would have reversed the base sequences, right? So that's the reason why we have a fixed reading direction for mRNA. Now the eighth characteristic, the code has a start and stop signal which specifies the start, that is initiation of protein synthesis, and termination of protein synthesis, that is stop signals. So they are present in both prokaryotes and eukaryotes and they are universal. So you can see here AUG is always the start codon for protein synthesis. Okay, so this is the start codon. So there are only 61 of 64 codons which specifies for amino acids. Okay, why? Because there are other three codons which is UAG amber, UUA opal and UGA ochre. They do not code for any amino acid and that's the reason why they are known as nonsense codon or stop codons, and the one which specifies they are known as sense codons. Okay, so they make any sense, right? They are coding for an amino acid, so they are termed as sense codon, and one all means all these three codons they do not specify an amino acid, thus they are termed as nonsense codons or stop codons. So I guess now all these 8 characteristics are clear to you. Let's move towards Wobble hypothesis. So wobble, it actually means unsteady movement. Okay, let's see how this term is applied here. So wobble occurs in anticodons. Okay, now here we are talking about anticodons, not about the codons on mRNA. Okay, we are talking about anticodons, and where are anticodons present? They are present on tRNA, that is transfer RNA, which brings the amino acid to the mRNA for coding. Okay, now the Wobble base pairing or the Wobble rule, it explains how the genetic code is translated from DNA or RNA into amino acids during the protein synthesis. So it was proposed by Francis Crick in 1966. Okay, so this is the Wobble base pairing. Let's see in detail. So in protein synthesis, we know that a set of 3 consecutive nucleotides in mRNA, they are called as codon. Each codon codes for specific amino acid. We have learned this as well. There are 64 possible codons or 64 possible codons which codes for 20 amino acids, standard 20 amino acids, of which 61 are the sense codon and 3 are the non-sense codons, right? We have revised this. Now, this means that multiple coding can happen. That means multiple codons can code for a same amino acid. Okay, let's see. So you can see here multiple codon can code for a specific amino acid. For example, threonine is the amino acid. So you can see here 1, 2, 3, 4. 4 different codons can code for threonine. You can see here for leucine how many codons can code. 1, 2, 3, 4, 5, 6 codons can code for leucine. For phenylalanine there are 2 codons. Okay, so you can also see here we have 3 stop codons and we have a start codon as well. Okay, so that is how this table is used to know which code codes for a particular amino acid, because it is difficult for us to remember all the codons and all the amino acid they code for. Okay, so let's see what the Wobble hypothesis states. Before that, this is how the normal pairing happens. Okay, this is the codon on mRNA and this is the anticodon on tRNA. So you can see here this is the tRNA which is carrying a amino acid leucine, and this is the anticodon GAG. Okay, so let's check. So GAG, CUC is the codon. So C, this is the U, so CUC, it will code for leucine. Okay, so always this table is studied or referred when you are referring to the codons on mRNA and not the anticodons. Okay, so the basics like purines and pyrimidines, they form the base pairing. Okay, so you know that guanine codes, oh sorry, not codes, it pairs with cytosine, and adenine will pair with uracil because there is no thymine in mRNA, right? Then G will again base pair with C, that is guanine will base pair with cytosine. This is the normal pairing, but in case of Wobble pairing, the third nucleotide in the codon, so first is C, second is U, and again the third pair here is U. Okay, so this is flexible for pairing with the anticodon, third code of the anticodon. Okay, sorry, first nucleotide of the anticodon, because this is the 5' end and here is the 5' end of mRNA. So for mRNA, C is the first nucleotide, but for tRNA anticodon, G is the first nucleotide. So the pairing is of third nucleotide of mRNA and first nucleotide of anticodon of tRNA. Okay, so the third base pair, the third base of the mRNA is flexible for pairing with the first base on the tRNA. Okay, that is Wobble pairing. Now the Wobble hypothesis states that the third position of mRNA codon is less constrained in base pairing. That means it is less natural in base pairing with the corresponding anticodon of the tRNA during translation. Okay, the anticodon is the sequence of 3 nucleotides in the transfer RNA, that is tRNA, right? Transfer RNA is this, and it is having a codon, but we term it as anticodon because it is complementary to the mRNA. Now the first two positions of the codon anticodon interaction, they follow strict base pairing rule, that is adenine to uracil, then uracil to adenine, guanine to cytosine, and cytosine to guanine. But the third position of anticodon codon pair is more flexible. Okay, and that's the reason why Wobble is possible in anticodons. Now this flexibility, it allows certain tRNA to recognize multiple codons. For example, here now in this particular image you will see that there is I written over the anticodons, okay, not in mRNA. I'm talking about the anticodon on tRNA. So this I stands for inosine, and it actually pairs with adenine, cytosine and uracil, but not with guanine. Okay, so remember that you will, I don't know if you have this in your syllabus or not, but if you have, then you will study this in detail. I'm just referring here that in some tRNA you will find that anticodon has I, that stands for inosine. Okay, remember that. Now this is this flexibility of the third base and the first base of anticodon base pairing, it allows certain tRNA to recognize multiple codes that differ only in the third position while still carrying the same amino acid. This phenomenon is known as wobble. Okay, so for example, you have GGU, this is the first case, GGU, then GGC second, then GGA third, and GGG is the fourth. Now, for all these four codes on the mRNA, which codes for glycine, single tRNA will carry the anticodon, that is CCA, and they can and this particular tRNA can recognize all these four codons. Okay, we don't need four different tRNAs here. Why? This is because of the Wobble. You can see here now there is CCE on the anticodon, but here you will find that there is CCI because inosine can also pair with uracil. We again have CCI because inosine can pair with cytosine. Again we have IIC because inosine can pair with adenine, but here we have CCC because inosine doesn't pair with guanine. Okay, remember this basic. Then CCC is why, because cytosine pairs with the guanine. Now this is the single tRNA which will or which can read this code on mRNA, and it will code for glycine. You can see here glycine is present on this particular tRNA. Okay, so this tRNA will carry glycine if in sequence there are all these four codes present, so there will be four glycines in the polypeptide chain. Okay, so this is Wobble hypothesis. Now the Wobble hypothesis increases the efficiency and the flexibility of protein synthesis as it reduces the number of tRNA molecules required for translation and allows cells to produce proteins more rapidly and economically. Okay, so why there is Wobble hypothesis? Because the cell it reduces the number of tRNA molecules required for translation and thus allows to produce protein more rapidly and economically. That's the reason why Wobble hypothesis is important, and that is the reason why we study in our theory. Okay, so last slide of this video. So we will just revise how to read this codon table, and we will try to find out these two codons code for which amino acid. Okay, so first letter we have here, we have to refer the row, then second letter we have in columns. Okay, again third letter we have in row. So that is how we refer the codon table. So first letter is U, so we will see this particular row. Then second letter is again U, so this row and this column, and third letter is G, so here it is G, so it codes for UUG, codes for leucine. Okay, so UUG codes for leucine. Now second is CCA, so first letter is C, so this particular row, second letter is again C, so this column now. Okay, and third letter is A, so it codes for, you can see here, here it is A, so it codes for proline. Okay, so this particular codon codes for proline. So that is how we use genetic codes and Wobble hypothesis in translation, and then you can find out which particular code in the given sequence codes for which amino acid, and even you will get these questions to solve in your molecular biology biochemistry papers. Okay, so get your concepts clear, do watch these videos, do like my videos, do share these videos with your friends, and do subscribe to my channel. Thank you.